viewers, today I will discuss about the classification and biological significance of enzyme classification and nomenclature. Enzymes are classified into 6 different groups, but today I will talk about another one group as well according to the reaction being catalyzed. The nomenclature was determined by the Enzyme Commission in 1961, hence all enzymes are assigned as EC number, Enzyme Commission number. The International Union of Biochemistry and Molecular Biology had developed this nomenclature. Enzymes are generally named according to the reaction they catalyze or by suffixing "ase" after the name of the substrate. Each enzyme is described by a sequence of 4 numbers preceded by EC. EC denotes Enzyme Commission and the number of the enzyme is called EC number. There are so many different names of one enzyme. Say for example, the ATP plus creatine can be transformed to ADP plus phosphocreatine by one enzyme. The colloquial term general name is creatine kinase, but systemic name is ATP is to creatine phosphotransferase EC 2.7.3.2. So, the general principle must be there. The name of the enzyme will be accepted universally. The first general principle of this is that names purporting to be names of enzymes, especially those ending in "ase", should be used for single enzymes. The second general principle is that enzymes are primarily classified and named according to the reaction they catalyze. A third general principle is adopted that the enzymes are divided into groups on the basis of the type of reaction catalyzed. Scheme for the classification of enzymes and the generation of EC numbers. The first number shows to which of the 6 main divisions, 6 or 7 rather, classes the enzyme which it belongs. The second figure indicates the subclass. The third figure gives the subclass, and the fourth figure is the serial number of the enzyme in its subclass. So, main divisions, actually there were 6 divisions, nowadays the 7th one is incorporated. Number 1 oxidoreductase, number 2 transferase, number 3 hydrolase, number 4 lyase, number 5 isomerase, number 6 ligase and number 7 translocase. Within oxidoreductase we can find dehydrogenase, oxidases, reductase, peroxidase, catalase, etc. Within hydrolases, esterase, glycosylase, peptidase, phosphatases are there, phospholipases as well. Within isomerase, racemase, epimerase, etc. are present. Within transferase, transaldolase, transketolase, kinases are present. Within lyases, decarboxylases, aldolase, hydratases are present. And within ligases, synthetase and carboxylases are present. First one, oxidoreductase. These particular enzyme catalyze oxidation reduction reaction. The substrate is regarded as hydrogen donor and it is getting oxidized. The systemic name is based on the donor is to acceptor oxidoreductase. Dehydrogenases and oxidase belong in this category. Say for example EC 1.1.1, here the donor is CHOH and NAD plus is acceptor. It is for alcohol dehydrogenase. EC 1.1.2 is for lactate dehydrogenase where the donor is CHOH and the cytochrome is acceptor. Number 2, transferase. Enzyme catalyzes the transfer of a functional group that may be methyl or phosphate from one molecular compound that acts as donor to other acts as acceptor. EC 2.1, methyl group transfer happening here like methyl transferase. EC 2.2, transketolase, ketone group is transferred. Number 3, hydrolase. It catalyzes the hydrolysis of chemical bond. Hydrolytic cleavage of CO or CN bonds happen. EC 3.1 is responsible for esterase. EC 3.2 is responsible to hydrolyze sugars, glycosylases. Number 4, lyase. It catalyzes the breaking of various chemical bonds by means of hydrolysis and oxidation. EC 4.1 cleaves CC bond, decarboxylase. EC 4.3 cleaves CO bond in dehydrolyase. Isomerase. It catalyzes the structural rearrangement of isomers, geometric or structural change. It includes tautomerase, epimerases, etc. EC 5.1, D-alanine racemase. EC 5.2, cis-trans isomerase. Number 6, ligase. It catalyzes the joining of two large molecules by forming new chemical bonds, accompanying the hydrolysis of diphosphate bond in ATP. EC 6.1, amino acid tRNA synthetase, it ligates CO bond. EC 6.2, CS bond ligates, malate-CoA ligase. Translocase, this one is currently incorporated here. It catalyzes the movement of ions and molecules across the membranes or their separation within the membranes. The transporters are included here. Difference between synthase and synthetase. Synthase catalyzes a process without ATP and actually it belongs to under lyase. The example is HMG-CoA synthase, ATP synthase. But synthetase, it requires ATP for its action and comes under ligase. The example is succinyl-CoA synthetase, amino acid tRNA synthetase rather. The biological functions, right? Now, I will discuss about the importance of the enzyme in biological, medical and other aspects. We know that enzymes are needed for signal transduction and cell regulation process. Kinases can phosphorylate some downstream protein and phosphatases eliminate the phosphate groups. In this way, they can regulate the signal transduction cascades. The generation of movements, muscle contraction, movement of cytoskeleton, various ATPases are needed. And we know for active transport, sodium, potassium, ATPases are needed which act as ion pump. Luciferase enzyme, it generates light by firefly. In case of virus, for infecting cell, HIV integrase is needed. Reverse transcriptase is needed. So that they actually help to integrate their genetic material. For viral release, neuraminidase is needed for influenza virus. For digestive purpose, we have amylase, protease, cellulase, etc. For ruminants, microorganism, they actually synthesize cellulase. Control metabolism. Say for example, we need to phosphorylate glucose by glycolysis, we need the help of hexokinase, phosphofructokinase as well. So, when we are talking about the biological role, the protein kinase and phosphatase, they regulate signal transduction, and to regulate metabolism, hexokinase as well as phosphofructokinase is there. Now, I will talk about the importance of enzyme regarding industrial application, biological functions, medical importance, clinical importance, including involvement in diseases. Number 1, involvement in disease. Mutation of some gene may lead to the formation of some abnormal enzymes which may cause the malfunctioning. In case of phenylketonuria, there is the mutation of the genes for the enzyme phenylalanine hydroxylase which converts phenylalanine to the tyrosine, right? If it is malfunction, so phenylalanine is getting accumulated causing mental retardation. Germline mutation of UV-related repair enzymes can cause the xeroderma pigmentosum, a hereditary syndrome causing skin cancers. Industrial application of enzymes, one by one I will tell about them. Number 1, papain is needed for wine manufacturing. For cheese making, rennin is needed. Lipase adds flavor to it. Candy making, invertase is needed. Clarifying fruit juice, pectinase is needed. To tenderize meat, papain is needed which breaks down the collagen helices against hydroxyproline residues. For bread whitening, lipooxygenase is needed. Distain fabrics, alkalase. Recovering silver, pepsin. Correcting digestion, in vitro amylase. For wound healing, proteolytic enzyme trypsin is needed. Analyzing biochemicals, uricase and urease. To dissolve blood clots, urokinase. Augmenting surgery, trypsin. Breaking down chemicals, monoamine oxidase. It allows brain to communicate. Alpha amylase is needed to starch breakdown. For rubber industry, catalase, it turns latex to foam. Beta galactosidase, it hydrolyzes lactose which is used in dairy industry. For medical application, L-asparginase, it removes asparagine from the tissues needed for tumor growth, right? So, within the tumor cells, if asparaginase is given, asparagine can be moved which may not serve as food for the tumors. So, it is the leukemia therapy used. Urokinase removes blood clots in heart diseases. Glucose or oxidase, it is needed for in vitro blood glucose estimation. So, it is called GOD-PAP method. It is kid-based method where with the help of glucose oxidase GOD, glucose is oxidized to gluconic acid by the help of oxygen and H2O2 is produced. These H2O2 along with phenol and 4-amino antipyrine with the help of peroxidase produces quinonoid dye, which is measured by spectrophotometer, and the OD value at 532 nanometer of this quinonoid dye is proportional to the blood glucose concentration. Clinical application. The enzymes are needed for therapeutics, for clinical analysis like cholesterol oxidase for cholesterol, triglyceride, lipase analysis, disease diagnosis, protease, pronase, RNase, they are needed for wound healing. Streptokinase from streptococcus, it is needed for clearing blood clots to myocardial infarction. So, from plasminogen pre-enzyme, the plasmin activated serine proteases are produced which cleaves fibrin to the soluble component. In research, the enzymes are needed. Say for example, lysozyme which breaks 1,4-glycosidic bonds, the source is hen egg white, is needed to disrupt bacterial cell wall. Endonucleases, restriction enzymes, are needed to break down DNA fragments. The source may be the bacteria, right? EcoR1, MSC1, so many restriction enzymes are there, which is used for recombinant DNA technology, gene manipulation and DNA fingerprinting. Analytical application, glucose oxidase that I have already told, it oxidizes glucose and needed for the test for the blood glucose analysis. The source is fungi. Luciferase produces light. It is obtained from fireflies and used to detect bacterial contamination in food material. Other application, alpha amylase breaks down starch. Source is bacteria which converts starch to glucose needed for food industry. Glucose isomerase converts glucose to fructose. Source is fungi. It is needed for high fructose syrup production. Proteases which digest protein. Source bacteria needed in case of washing powder. Renin clots milk protein. The source is animal stomach and needed for cheese making. Catalase converts H2O2 to H2O as well as O2. It is extracted from animal livers. Once again, it is needed in case of rubber industry to convert latex to foam. Beta-galactosidase is needed to hydrolyze of the lactose sugar. The source is fungi and needed for the dairy industry. Antioxidant enzymes, superoxide dismutase, catalase, glutathione peroxidase as well as glutathione reductase, they are needed to scavenge superoxide as well as so many other oxyradicals produced during the course of oxidative stress in cell. There are some non-vitamin part along with the enzyme, they can act as coenzyme. S-adenosyl methionine which transfers methyl group and it is found in bacteria and eukaryote. Coenzyme B which transfers electron found in methanogens. Coenzyme M transfers methyl group are also found in methanogens. Heme group, it transfers electrons, are found in bacteria as well as eukaryotes. Multi-enzyme system. Here some enzymes they are acting in the mosaic fashion. The product of one enzyme can act as a substrate for other. In the case of E. coli, pyruvate dehydrogenase complex, we can find three enzymes. Number 1, pyruvate dehydrogenase. Number 2, dihydrolipoamide acyl transferase as well as dihydrolipoyl dehydrogenase. They actually act together to give rise to the acetyl-CoA from pyruvate as well as NADH is getting produced. The evidence of the ES complex. The change in fluorescent intensity in case of tryptophan synthetase which converts serine plus indole to tryptophan can be monitored easily via change in fluorescence intensity with time. So, if you follow the fluorescent intensity at a particular wavelength, enzyme has a basal level of the intensity due to the presence of the coenzyme NADH. Now, when it binds to the first substrate serine, there is a hike in the fluorescent intensity. But after binding to the second substrate indole, the quenching happens below the basal level. Now, when the product is getting formed as tryptophan, the enzyme goes back to its original fluorescent intensity. The evidence of ES covalent complex is found in case of fructose biphosphate aldolase. So, we can find that it has lysine residue at its active site which is actually derivatized by the help of borohydride. The substrate specificity of the enzyme actually indicate the characteristics of the substrate. So, it has some susceptible chemical bond as well as it must have some positioning group. In case of acetylcholinesterase, in acetylcholine we must find the binding of or the positioning group, that is quaternary amine along with the susceptible bond. In case of chymotrypsin as well, along with hydrophobic positioning group, the susceptible bond is present. So that's why it is called hydrophobic acyl group transferase. The affinity labeling to know the active site amino acid, the chymotrypsin can be treated with TPCK, that is N-tosyl L-phenyl alanyl chloromethyl ketone. Enzyme and substrate in living cells. Some enzymes are needed in high concentration, some are in low concentration. Hexokinase, it reacts as at central metabolic pathway to phosphorylated glucose. So concentration is 100 micromolar. But enzyme which is needed for biosynthesis of HMG-CoA synthase enzyme, so it is needed for the biosynthesis of some coenzymes, it is not needed so much. The concentration is very low, 0.01 micromolar. The substrates are not always sufficient in cells to saturate the enzymes. Enzyme activity, some parameters must be taken care of. That is the stoichiometry or concentration of the substrates, requirement of the cofactors, as well as substrate and cofactor concentration or affinity correlated with the Michaelis constant Km, pH optimum. And we know that initial rate of reaction is proportional to the enzyme concentration. The temperature optima, because enzyme must be stable. Simple analytical procedure must be adopted for the quantitative estimation of the enzyme activity. So we will discuss about some of the assays. Enzyme assay. Enzyme assays are the lab methods for measuring enzyme activity. They are vital for the study of enzyme kinetics as well as regulation and inhibition. So we can measure either the consumption of substrate or product formation with time. Broad classification includes continuous as well as discontinuous process. Continuous spectrophotometric assay may be direct or coupled. So rate of reaction can be increased with the enzyme concentration. Enzyme activity can be measured. So if we follow the change in the absorbance with time divided by molar extinction coefficient, so we can get U per ml, that is the activity of the enzyme. So activity of enzyme is equal to rate into reaction volume. Types of assay may include initial rate experiment, progress curve experiment, transient kinetics experiment or relaxation experiments. Now there are so many factors which may control the assay. Salt concentration, usually it is in between 1 to 500 millimolar. Effects of temperature, because temperature optima must be maintained, it is around 37 to 40 degrees centigrade, but some hot spring belonging bacteria, they can endure 100 degrees centigrade even. Effects of pH, otherwise there must be the distortion of the ionic structure within the enzyme active site amino acids. Substrate saturation, so substrate saturates all the active site of the enzyme, it will go to the Vmax, maximum velocity. It will follow the zero order kinetics. The level of crowding, that is macromolecular crowding, can change the rate of reaction as well as equilibrium. Now, first I will talk about the continuous assay. The assay which give the rate of reaction with no further work necessary. Continuously the activity or rate of reaction can be monitored. It may be spectrophotometric. It may be fluorometric. The sample can be prepared, can be fixes, and absorbance can be monitored at specific wavelength. It is more specific spectropluorometric study than that of spectrophotometric study. Calorimetric study. So the heat change during the course of enzymatic reaction can be measured by microcalorimeter. Chemiluminescence, the Horst redox peroxidase action can be monitored by change in the color. Okay, so that is actually done in case of ELISA analysis. Light scattering can also give the idea about the enzyme activity within some time course. The assays, first continuous assay based on the spectrophotometric, either it may be the colorimetric change or maybe the change of the substrate or product within the UV wavelength. So colorimetric change can be observed by MTT assay, tetrazolium dye is a substrate here, or sometimes the activity of the alkaline phosphatase can also be monitored by means of paranitrophenyl phosphate, and paranitrophenol is produced which is yellow in color and can be monitored at the wavelength 405 nanometer. In the direct assay, in case of lactate dehydrogenase, where from lactate to pyruvate is produced, simultaneously NAD plus is converted to NADH. Now these NADH has absorption at UV range 340 nanometer. So if you find the increase in the absorbance of product, that is NADH, it will give rise with respect to the time the activity of the enzyme. Now coupled assay. So direct assay means we can measure directly the absorbance of the substrate or the product. Sometimes neither substrate nor product may have the absorption within the visible or UV range. Say for example in case of pyruvate kinase, which actually transfers phosphoenol pyruvate and ADP to pyruvate and ATP. Neither substrate or nor product has absorption. So these pyruvate kinase activity or reaction is coupled with once again lactate dehydrogenase reversible reaction, where pyruvate is transformed to lactate and NADH is utilized to NAD+. So as NADH is utilized, so the utilization can be monitored by decrease in the OD or absorbance at 340 nanometer. So within some given time we can measure the activity of the enzyme by coupling this reaction. Discontinuous assay. Samples are taken from enzyme reactions at intervals time to time, and then amount of the product formation is measured, not continuously. For discontinuous assay process, it includes radiometric assay, where the scintillation counter is used, chromatographic assay, which includes HPLC, high performance liquid chromatography assay. Control of enzyme activity. The enzyme activity can be controlled, enzyme production can be enhanced or diminished at the level of transcription or translation by a cell in response to the changes in the cell environment. Enzymes can be compartmentalized with different metabolic pathways occurring in different cellular compartments. We know that the effect of the polyphenol oxidase, it is compartmentalized usually. Fatty acids, they are getting synthesized in endoplasmic reticulum as well as Golgi, but they are utilized in mitochondria. Enzymes can be regulated by inhibitors and activators. We know that last product can inhibit the committed step in a series of metabolic reactions. Enzymes can be regulated through covalent modulation. So palmitoylation, phosphorylation, it can regulate the enzymatic activity. We note that in case of glycogen synthase, phosphorylase, they can be activated or deactivated by phosphorylase or phosphorylation by kinase, as well as removal of the phosphate group by the phosphatases with respect to the insulin hormone. Some enzymes may become activated when localized to a different environment. We know that there is a conformational change of the heme agglutinin of influenza virus. So the conformational changes can only be happened when it present in acidic environment of lysosome. Enzyme regulation. The amount of the enzyme synthesized either by the induction or repression can be regulated. Positive and negative modulators by activators and inhibitors. Covalent control. One enzyme can control other enzymes, say for example kinase and phosphatase can control the downstream enzymes. Phosphorylase kinase can control the phosphorylase in case of glycogen breakdown. Product and substrate ratio, that is mass action. Multiple forms of enzymes or isozymes. Regulation of metabolic pathways as well as regulatory site by concomitant step. Cellular enzyme concentration that happens in case of glucose and pyruvate by carboxykinase response in blood. The Enzyme Unit U was adopted by the International Union of Biochemistry in 1964. The unit CATAL, recommended by the General Conference on Weights and Measures in 1978 and officially adopted in 1999. Enzyme units U. It is the amount of enzyme which causes transformation of 1 micromole of substrate per minute at 25 degrees centigrade under optimal condition. CATAL, the amount of enzyme activity that transforms 1 mole per second of substrate. 1 U is equal to 16.67 nanocatal. Specific activity. It is the number of enzyme unit per mg of total protein. It is the indicator of enzyme purity. It increases during enzyme purification and maximum and constant at pure state. Molar or molecular activity or turnover number. It is the number of substrate molecules transformed per minute by a single enzyme active site. For carbonic anhydrase, it is 36 into 10 to the power of 6 per minute per molecule. Catalytic power, that is catalyzed rate divided by uncatalyzed rate. The rate of a reaction is the concentration of substrate disappearing divided by product produced or the product produced per unit time. that is mole per liter per second. The percent purity is the specific activity of enzyme sample divided by specific activity of pure enzyme into 100. Enzyme activity can also be given as that of certain standard unit of substrates such as gelatin, then measured in gelatin digesting units GDU, or milk proteins that measured in milk clotting unit or MCU. So students, I have discussed about the grouping, classification, nomenclature of the enzymes, biological, clinical, medical as well as industrial significance, enzyme assay, unit, and some points regarding enzyme specificity, regulation, control, etc. Thank you.